Okay, good evening to all. I'm Dr. Pankaj from JSS College of Pharmacy, UP, also convener for Scientific Committee, PhamaNEST21, which is organized by Pharmacy Federation. So today is the last day of poster presentations. Since last 1 week, we are completed many presentations. So I welcome today's evaluators for this presentation, Dr. Subhas, Dean School of Pharmacy, GH Raisoni University, Madhya Pradesh. Welcome you, sir. Dr.
Thank you, sir.
Madhav Dwivedi
Yes,
from
sir.
Gitanjali University, Udaipur. Welcome you, sir.
Thank you, sir.
Dr. Shankar, Professor, PhD College of Pharmacy, Coimbatore, Tamil Nadu. So we welcome you all. And let us start the session. Susa.
So now I request poster number PhamaNEST 456 to kindly come up and start presenting the poster. Also, I request Chetan to kindly change the poster to 456.
Hello, am I audible?
Yes, you are audible.
Yes. Thank you so much. Thank you, PhamaNEST, first of all, to give us an opportunity, the young researchers, to have a platform like this is a very good thing for us. Good evening, everyone. Good evening, guests, and good evening, all the students and delegates who are over here. I would like to present my research work, which we performed in our VIT or University Vellore at the time of our PhD, which was related to the bioinorganic chemistry. And the topic of today's e-poster presentation from my side is DNA binding behavior of mixed ligand vanadium 5 complex based on novel tridentate hydrazone moiety and another ancillary ligand benzene hydrazonic acid. Means how the DNA binding behavior of vanadium complex with one novel hydrazone ligand and one ancillary ligand will be actually exhibited. So we have tried to explore the same thing. I would like to go ahead with the introduction as why we have chosen the vanadium in this case, because vanadium being a very important biologically essential metal ion which is required for different kind of biochemical processes, and as we know already, DNA binding agents, DNA interacting agents, and DNA intercalators like cisplatinins are already in the market since long period of time, which are active against specifically with the against very specific tumors. And due to the toxicity of the platinum containing drugs, we medicinal in our bioinorganic chemists have actually taken interest for the designing of the different types of metal complexes with different metal cores. So in the meantime, since 10 years, ruthenium metal complexes, cobalt, copper, as well as zinc metal complexes also have come in the picture of and we have seen them acting as an anti-cancer agent and with the different types of mechanistic pathways. So anti-cancer agents do have different type of mechanistic pathways in order to kill the cancerous cells. They do effect on the cell signaling pathways. They do inhibit the cell DNA polymerization. They do even inhibit the cell growth at specific cell cycles. We are herein going to explore the anti-cancer effect of the metal complex of the vanadium by how it is interacting to the DNA and how that DNA interaction of that vanadium complex can be converted into the cytotoxic activity. So in the literature, we actually reviewed different types of vanadium complexes which were already reported. Most of the times, vanadium 4 has been reported since some 10 to 15 years because it is easy to prepare and they are very small molecules which can get easily inserted into the DNA base pairs. But we wanted to try it further and to give a new try about how the vanadium 5 complex will react, and if we change the different ligand system and if we create kind of a bit planar molecule, will it be able to even interact to DNA properly, and will it be really acting as a cytotoxic agent? So we have designed hydrazone based tridentate hydrazone based ligand based on the 4-chlorobenz hydrazide, which was treated with the diacetone derivatives, methyl phenyl diacetone derivative, and then we have come up with one hydrazone ligand which has tridentate binding system. And then we considered about the valency of the vanadium 5 complex. So we needed two more dentate sites in order to complete its 5 valency, and therefore we, instead of taking phenanthrene, instead of taking 110 phenanthroline, instead of taking bipyridine, the settled one ligands, bi-ligand system, we have tried enough. We thought of taking benzhydrosomic acid as one of the ancillary ligand. So in this whole work, I would just like to give you the outline. We tried to synthesize one hydrazone ligand which was novel. Then we considered and benzhydrosomic acid as an ancillary ligand, and then we synthesized vanadium 5 complex on the basis of these two ligands. The complex was quite planar in the nature, as it was having 3 aromatic rings in the same structure itself. Then we tried to assess the DNA binding interaction or DNA binding mode of the same complex by using different spectroscopic techniques, and considering those DNA binding efficacy of that metal complex, we compare the same we perform the same spectral analysis for cisplatin as well. So in order to understand how much our complex is really getting inserted into the DNA base pairs, we compared our DNA interaction spectroscopic techniques with the spectroscopic studies with the cisplatin as well. And at the end, we performed the cytotoxic studies on the MCF7 breast cancer cell line. So here is the synthetic scheme. We synthesized the hydrazone ligand in the presence of acetic acid, the hydrazide, 4-chlorobenzhydrazide and diphenyl methylacetone in the ethanolic solution in the unimolar ratio was taken, and ethanol was the preferred medium of the solvent for carrying out the reaction. The ligands mass we have shown here very few characterization results, as we were not having much space in order to give all the data. So we have mentioned a prominent results of the characterization here. The hydrazone ligand was confirmed by using UV spectral analysis, FTIR, proton NMR, C13 NMR, and ESI and GCMS. So we have performed gas chromatogram for the hydrazone ligand, and we received single peak at the retention time 21.77 minutes. Then we also have performed then after having the pure ligand or 99% of the purity of the hydrazone ligand, we went ahead with the reaction and we performed synthesis of the vanadium 5 complex. By considering the dentate sites, we have taken 1 mole of the hydrazone ligand, 1 mole of the benzhydrosomic acid, and 1 mole of the vanadial acetylacetone. Being vanadial acetylacetone is the source of the vanadium to get incorporated into the different ligand systems, and therefore we expected the structure of the ligand in the similar way, like there should be vanadium in the center, which is having hydrazone ligand along with the benzhydrosomic ligand attached to the same. So we carried out the reaction in the methanolic solutions with the all reagents were in the equimolar ratio, and we performed the reflux condensation. We received greenish color compound, and then we purified it by using non-polar solvents like ether, so that any other impurities, if those might be present, will be going away. And then we carried out the ESI mass for the metal complex. Even we have carried out UV analysis of the metal complex, FTIR analysis of the metal complex. Here, I would like to explain about the UV spectral details of the ligand, hydrazone, benzhydrosomic acid, and the vanadium complex. If we can see here, these are the basic ultraviolet spectral studies of them. As ligand, hydrazone ligand coordinates with the vanadium 5, there is the electronic transitions between, or you can say ligand to metal transfer will be there, and therefore we can observe the extra peak at 455 in the case of the complex, where that peak is absent in the case of the ligands, because those ligands are organic in the nature. We can't observe those metal to ligand charge transfer in the case of the ligands, and whereas we have observed that transition in the case of the complex, where we confirm that the coordination might have happened. And even we confirm the coordination behavior with the FTIR spectral details as well, we observed that the carbonyl group of the hydrazone ligand is taking part in coordination with the vanadium metal directly, which is causing the shifting of the wave number of the C double bond O in the ligand towards the lower wave number after coordinating to the metal ion. And at the end, we even have as we needed to perform the biological studies, in vitro biological studies, we needed to confirm the molecular weight of the metal complex, and we performed ESI mass studies for the same. We dissolved the complex into as we have received it from the methanol, we actually crystallized in the amorphous way, not in the exactly single crystal. So we received this ESI mass at with the MBAS Z value at pi 69.94. So after confirming the structural coordination behavior and its molecular weight and its spectral important wavelengths and wave numbers, we went ahead with the in vitro DNA binding activities. So to ascertain the mode of binding to the DNA of the complex, first of all, we have performed the UV titration. So UV spectral studies of the complex with the DNA were performed. In the successive increments of the DNA were added, we kept the concentration of the complex in the cuvette constant at 25 micromolar, prepared in the tris HCl buffer, and even we, before going ahead or before mixing the solution of the complex with the buffer, we even analyze the stability of the complex in the tris HCl buffer at least for 72 hours. So we observed that vanadium metal complex is not getting dissociated even after 72 hours through the UV spectral studies, and it is not going to react with any kind of ions which might be present in the buffer. And then we went ahead with the UV spectral titration studies of the DNA. What did we observe? We kept the concentration of the complex constant, and we kept on increasing the DNA's concentration by at least we performed from increments were from 0 to 140 micromolar, and we observed a little bit of red shift as well as hypochromicity. So when there is a red shift observed, means increase in the wavelength after each successive increment and decrease in the absorbance in the specific spectra, we had a chance that this mental complex might be having an intercalative behavior. So what did we thought? Okay, if this complex is also intercalating, why not to compare the same? Why not to compare with the standard intercalator we have in the market, like cisplatin? So we performed even UV spectral titration with the same DNA, with the same concentration of the cisplatin 25 micromolar as of the complex, and we received this Kb, that is binding constants to the UV spectral studies. We observed our complexes binding constant was 3.84 into 10 raised to 4 molar inverse, whereas we received cisplatins ones as 4.27, which were nearly equal to each other. And then to further ascertain and actually having an idea whether it's really intercalating or it's just substituting the best pairs, we went ahead with the quenching, fluorescence quenching assays with two specific dyes, ethidium bromide and acridine orange. Ethidium bromide is a classical intercalator which gets inserted between two base pairs of the DNA, and it enhances the fluorescence, and therefore we can observe those ethidium bromide DNA complex under the fluorescence spectrophotometer. We took the same kind of principle with us, and then we started again fluorescence spectral titrations in the presence of ETBR DNA solution. So ETBR DNA solution was kept constant, and we kept on adding the successive increments of the complex in the same, and then we observed that there is a quenching. So we observed 42% of the quenching in the case of metal complex, vanadium, and 46% of the quenching pattern in the case of the cisplatin. Then what we went ahead, we really wanted to know whether, because even groove binders are also known to be quench ETBR, means they may displace ETBR from any period of time, any side, and they can just bind to the groove as well. But we wanted to more confirm the thing about the intercalative behavior of this planar vanadium complex. So we went ahead with the acridine orange. Acridine orange is a dye which reacts with the base pairs.
Sorry to interrupt. Can you be just come to a conclusion, because you are explaining all the methods.
Okay, sir. Okay, sir. Okay.
Tell
Yes,
what is what is your?
So, in the case of the acridine orange, we observed that acridine orange reacts with the adenine and thymine, and then we get the staining, we get the quenching in that case, and we observed that even complexes replacing that dye. In the case of the circular dichromosome studies, we incubated the metal complex along with the cisplatin, and we observed that our complex is intercalating with the DNA and it is affecting the helicity and the conformation. In the case of the viscosity, as after the intercalation, DNA's length increases and that causes change in the viscosity. In order to confirm the simulation behavior, we have carried out molecular docking in 2D55 molar PDB-ID, and we observed that it is into the best space, it is intercalating. And in order, if it is intercalating, it is interacting to the DNA, we perform the cytotoxicity MTT assay on the MCF7 cell lines, and we observe the IC50 of vanadium 5 complex based on hydrazone ligand 6 micromolar and cisplatin 20 micromolar. Thank you so much. I finished within the time. I hope, sir, I have not disappointed you. Thank you so much.
Thank you, 456.
Yes,
Now
yes, yes.
I would like to request all the evaluators to please come up with their questions.
Sorry, because we are almost sitting.
Yes, sir. Yes, sir. Yes, sir.
Hours, okay?
It's fine, sir. No
Yeah,
issues.
so
We
our
can
aim-
understand. Being
our
working at home and managing the things is really difficult. We can
Okay.
understand, sir.
Fine. Yeah, from your presentation, I came to know that you want to develop a vanadium-based complexes, and you are compared with the cisplatin-based.
Yes, sir. Yes, sir. Yes, sir.
Yes, and you are telling that it is better than your cisplatin-based complex.
Yes, sir. This work got published in the Applied Organochemistry, Applied Metal Chemistry journal as well. Yes, sir. Thank you.
Okay. And how you thought that that is vanadium will be the choice for this one?
Sir, vanadium had been showing apoptotic activity earlier, vanadium 4 complexes.
Okay.
And we observed that in the cell signaling pathways, which are needed for the cell growth, vanadium actually acts on that signal, means other vanadium 4 and vanadium 3 complexes as well. So we considered about vanadium, why not to take the, let us take the vanadium but change its valency and change its coordination behavior, because we know that it has an apoptotic behavior already. So
whether compared to cytotoxicity, vanadium is very less cytotoxic, like that you are telling, and compared
yes,
to
sir, yes, sir, yes, sir. As even it is anti-diabetic potential was also unknown, so quite bioessential metal ion we can consider, because for the human beings we need some amount of the vanadium to be taken inside the body, like in the microgram level. So we considered taking the vanadium.
whether it can be used for targeting cancer drugs also.
Targeting cancer drugs means?
For carrier, as a carrier for.
Yes, even we can even prepare nanoparticles of the vanadium based on the polymers as well, and we can consider it as a targeted drug. We can devise actually in that. Yes, sir.
Up to what concentration it is not cytotoxic?
I guess we observed the IC50 at 6 micromolar. We received it kind of, we took it actually it is at very low concentration micromolar level, it is showing cytotoxic activity. So
Okay.
we have not gone below to that. We started the activity from 2.5, then 5, then 10, and then we till 20 mostly we have taken, because we were knowing already cisplatin has higher efficacy, and so we did not increase the concentration level while carrying out the cytotoxicity.
Up to 6 micromolar, it is the IC.
Yes, sir. Yes, sir. Yes, sir.
Okay, thank you.
Yeah. Thank you so much, sir. Yes.
Hello.
Hello, sir. Good evening.
You have told that you are changing the valency of vanadium.
Changing means we wanted to have a vanadium 5 complex, and considering tridentate ligand will give an idea of it will incorporate 3 dentate sites, or it can form a coordinate bond, and the ancillary ligand will form a hydrogen bond. So all over we wanted V double bond O, that will be considered as 1 valency, and other 4 will be taking part in the coordination behavior to complete it as 5. So because vanadium exists in 3, 4, 5 valency systems, so 5 is actually more stable considered complex system is vanadium 5.
So you have tried with all the three or only one?
In our PhD thesis, we tried even 4 and 5. But here in this research paper, we tried with the 5 as well. But the ligands are different in the case of oxovanadium 4. Here is, we can say, vanadium 5.
So you see the 5 is more better.
Because sir, better in the case of vanadium 4 actually even it depends on the ligands as well, and it depends on the valency as well. Being 5 as vanadium, if it is in the 5 valence state, there will be more number of coordination sites will be available and more number of ligands will be available. That will increase the planarity. But in the case of the 4, sometimes there are even small molecules can even small ligand molecules can be even taking part to form small vanadium 4 complexes. So that because of the more number of the valence system, there will be coordinating sites will be more, and that may increase the planarity, and that may actually give an better effect of the DNA interaction.
Okay.
Thank you, sir. Thank you so much.
No question from my side.
Oh, thank you so much, sir. Thank you, PhamaNEST.
Okay, sir. Thank you. Thank you, Poonam. Thank you, evaluators. Now I request.
Thank you.
Okay. Now I request Dr. Kalirajan to kindly give the vote of thanks.
Good evening. So I thank all the evaluators for your patient listening. So in our rules and regulation already we are given it's not more than 5 minutes and question, but some of the participants is not following. So so kindly excuse us. So I request Dr. B. Gaurama to conclude the remarks.
Okay, sir. A warm good evening to one.